Hi, my name's Henry. During this seminar, I'm going to be taking a look at the biotic stressor of sedentary endoparasites and their effects on plants. Okay, here we go. The phylum Nematoda, commonly known as roundworms, are amongst most ancient on the planet, estimated at around 1 billion years old. They are also one of the most diverse, with over 40,000 species estimated to exist, and present in almost every conceivable niche, and present at multiple trophic levels. As such, they play important roles in regulating population dynamics, soil nutrient cycling, and energy cycling, to name just a few. The focus of the seminar will be on the sedentary endoparasites. These are biotrophic parasites that spend the majority of their life cycle within a host. They establish permanent feeding sites in the vascular tissue in root cells, redirecting water and nutrients from the plant. This guild of nematode make up just a small minority of the total species, however are the focus of the bulk of the literature. This is due to the huge economic loss attributed to them through crop loss. It is estimated these parasites cost the agricultural industry over $100 billion annually. There are two genera of nematodes that I wanted to focus on. This is the root knot nematode and the cis nematode, because the bulk of this agricultural loss is due to them. Okay, this slide is going to be a very brief anatomy lesson, just to give you an idea of what these things look like. So when I refer to certain parts of their anatomy later on, you're going to know what I'm on about. The two most important features here are the stylet and the pharyngeal glands. The pharyngeal glands are where effectors are synthesized. These are a host of proteins that alter plant structure and function. I'll go into that in a little more detail later on. The stylet is the other important feature. It is functionally comparable to a syringe used to penetrate host cells and direct these secretions into specific cell compartments, like the cytosol or apoplast. Before we go into the main part of the seminar, I just wanted to give you an overview of the invasion process, so you can kind of visualize where you're at different points in the talk. It begins with the hatching of the nematode in its pre-parasitic juvenile form. After hatching, they immediately begin to migrate toward potential hosts. The mechanism for migration is not clearly understood, as this process is very difficult to monitor. However, it is thought to occur through chemical gradients of root diffusates, CO2 gradients, and temperature gradients. Once a potential host is reached, knot root and cis nematodes have unique modes of infection. Both begin by probing the root surface epidermal cells with their stylets, searching for an ideal entry point. These locations tend to be towards the elongation zone or meristem area at the tip of roots. Once found, root knot nematodes penetrates the epidermis and migrates intercellularly towards the root tip, where they enter the vascular cylinder. Here they establish a feeding site through the induced differentiation of 7 parenchyma cells and form large multinucleated giant cells. Once the feeding site is established, they can then mature into their adult forms. Cis nematodes, upon penetration of the root epidermal cells, migrate intracellularly through the root cortical cells towards the vascular bundle. Here they establish a feeding site through the fusing of adjacent root cells to form the multinucleated syncytia. Next slide. Oh, yeah. Both of these modes of parasitism require significant alterations to both plant cell structure and function. This is achieved primarily through the action of a diverse arsenal of effectors that alter the normal molecular functioning of plant cells. As mentioned previously, these proteins are primarily produced in the pharyngeal glands. These effectors are then secreted through the stylet in a very spatio-temporal specific manner, with certain effectors upregulated at certain times in the parasitic process. These effector molecules can change plant developmental processes, alter cell wall structure, And they can also modulate host stress and defense responses, which will be discussed in detail in assignment 3. The majority of the remainder of the seminar will be concerned with how nematodes affect plant cell structure and function. Okay, cell wall. This is the first barrier to the biotic stress of nematode parasitism. The plant cell wall is composed primarily of cellulose, but also hemicellulose, xyloglycan, pectin, and other proteoglycans. These structural components amalgamate to form a thick and rigid structure capable of withstanding significant pressure. In order to overcome this and facilitate successful migration, a range of enzymes and binding proteins are deployed that alter the architecture of the cell wall components. The first of these, the 1,4-endoglucanase, aka cellulase, was one of the first enzymes to be identified in root knot nematodes. Since then, the glycosyl hydrolase family has been identified in several species and is highly conserved. The cellulase acts by hydrolyzing the 1,4-beta-glycosidic bonds of cellulose, effectively breaking it down. This family of enzymes has also shown to have a similar hydrolytic activity for hemicellulose and xylin components, causing further cell wall degradation. Additional cell wall degrading enzymes have been identified, including pectin lyase, secreted by cis nematodes, and polygalacturonase, secreted by root knot nematodes. These both aid in the degradation of pectin. Additional effectors are also known to interact with the cell wall. Cellulose binding proteins that were once thought to have an enzymatic activity due to their cellulose binding domain, however, upon further investigation, were found to lack a catalytic domain. Functional assays confirmed an expansin-like activity that actually softens the cell walls by breaking the non-covalent bonds between cellulose strands. These cellular binding proteins actually mimic the activity of expansins that are naturally found in plants, usually used to promote cell wall softening to allow for cell growth. Taken together, these effectors act to significantly weaken the structural and functional integrity of the cell wall, allowing the migration of the nematode to its feeding site. Once a nematode reaches the vascular bundle, a series of functional and structural modifications are made to the plant root cells to establish feeding sites. Syncytia formation by cis nematodes is initiated when the selected root cell is punctured by the stylet. The cell is injected with a cocktail of effectors which lead to the cell dissolution and the fusing of adjacent cells, resulting in a multinucleated cell. A form of arrested mitosis is also stimulated in cells called endoreduplication. This is where cell cycle progression is stopped after DNA synthesis but before mitosis, resulting in elevated gene content. A similar process is observed in the formation of root knot nematode giant cell formation as acetokinetic mitosis, where cell cycle progression is arrested just after mitosis but before cytokinesis, resulting in two genetically identical nuclei. The exact mechanism by which the nematodes affect cell cycle is unknown. However, certain players have been identified. Studies investigating gene expression in giant cell and syncytia formation found high concentrations of cytokinin being expressed in both giant cells and syncytia. This was confusing at first because the phytohormone cytokinin has long been known to promote cell cycle progression, specifically aiding the transition from G2 to M and G1 to S phases. These are the phases also being inhibited at the nematode feeding sites. A recent study resolved this, however, by uncovering a concentration-dependent function of cytokinin at low concentration positively regulating cell cycle progression, and at high concentration negatively regulating it and promoting endoreduplication. The exact mechanism by which nematodes increase gene expression in nematode feeding sites is still unclear, However, it is thought to occur through nematode-derived transcription factors, altering host gene expression. Another mechanism potentially acting in concert with cytokinin and expression is a recently discovered effector, profilin-3. Profilins are actin binding proteins that sequester actin monomers. Its suggested role here is to trigger cell cycle arrest by rendering the cell incapable of forming a cytoskeleton due to a lack of available actin monomers, therefore inhibiting cytokinesis. It is likely there are a whole host of effectors at play here. However, the exact process still remains opaque. Okay. The stresses described so far have been quite localized in nature, operating on the molecular and cellular nature. However, once a feeding site is established, this changes. The nematodes effectively begin to act as metabolic sinks, drawing nutrients and water from the vascular tissue of the plant. This has a major effect on the rate of photosynthesis, as key nutrients in the synthesis of chlorophyll are used by the nematodes. A comparison study shows this by demonstrating that with an increased scale of infection, rate of photosynthesis decreases. The above-ground symptoms are very similar to those seen in drought and nutrient-deficient subjects, with plant wilting and stunted growth very apparent. Below ground, the roots undergo a very pronounced morphological change. Roots infected with cis nematodes are often dwarfed and have more frequent branches, whereas roots infected with root not nematodes tend to be more stubby and gooled. Anthropogenic-driven changes have the potential to dramatically alter nematode distribution and range. A study conducted into the potential range increase of a common virus-transmitting species projected an increase of 160 to 200 kilometers. Migration of virus-transmitting species and their subsequent introduction to native plant species could have devastating effects on plant populations, as they have no evolutionary history with these viruses. Likewise, this could lead to novel nematode-plant interactions, with plants lacking the stress response mechanisms to defend themselves. In a very similar vein, although not the topic of this seminar, in the case of animal parasitic nematodes, the co-invasion of foreign hosts and parasite into new ecosystems as a consequence of anthropogenic activity can lead to host switching. Native species unadapted to these foreign parasites experience an increase in parasitic virulence. Lastly, climate change may also make plants more susceptible to infection. Increased water stress due to drought, salinity stress, heat stress, all lower the plant's ability to mount an effective defense against invaders. This could potentially lead to higher infection rates in plants and potentially higher mortality rates as well. And the last slide is just my references. I hope you guys enjoyed my seminar on endoparasitic nematodes. Hope you learned a bit as well. Thanks very much, and yeah, have a good night.